Microbial diagnosis. Of diagnosis. The field of diagnostic microbiology determines whether suspected pathogen microorganisms are present in the test specimens collected from the human beings' environment and from the animals, if they are present in the test specimen or not. This diagnosis is used to isolate the microorganism and to identify the pathogen and can use it to treat the patients. The techniques used to diagnose the microorganisms are microscopy, culture techniques, biochemical reactions, cellological identification, and molecular biology techniques. Microscopy examination. First involves is the microscopy examination. Microorganisms can be examined microscopically for this staining techniques are used to identify the organisms. Stains we used are simple stain, gram stain, Gill-Nielsen staining, Giemsa stain, and spore stain. Simple stain. In this, take a slide and put the smear on it and apply it for the heat. Then add the methylene blue. After a few seconds, by washing with the distilled water, observe under the microscope. Then we can see the shape and type of the bacteria involved in the test specimen. Then the gram stain. On the slide, when we place the culture, then the primary stain, crystal violet, is added. After few seconds, modern iodine complex is added. Iodine complex is used to fix the crystal violet to the gram positive organisms. Then wash with the distilled water, so that the excess of crystal violet will be removed and the organisms can absorb the crystal violet. Then add the decolorization, which is the ethyl alcohol. When adding the ethyl alcohol, gram-negative bacteria, which does not absorb the crystal violet, will be free from the crystal violet. Then wash with the distilled water, so that the excess of ethyl alcohol will be removed. Then the secondary stain, known as the safranin, is used. By adding the safranin, gram-negative bacteria will obtain the pink color. Examples for the gram-positive are streptococci, staphylococci, monococci, and bacillus aureus and bacillus species. Whereas for the gram-negative, E. coli, salmonella, shigella, etc. are used. For the Gill-Nielsen staining, we apply the carbonyl fusion with heat on the slide containing the spear. Then we add the decolorization, which involves in the H2SO4 and ethyl alcohol by washing with the distilled water. Then we add the counter stain, methylene blue. By applying the carbonyl fusion and the acid-fast staining, acid-fast bacteria are in the pink color or red color, whereas the non-acid-fast bacteria are blue or purple color. Next involved is the Giemsa staining. Giemsa staining is mainly used to identify the malarial parasite. Giemsa staining, methanol and Giemsa stock is used. For the Giemsa stock, 250 ml of Giemsa powder is going to be dissolved in the 5 ml of glycerol. Then place it in the methanol. Then the Giemsa stock is prepared when the rest of 7 days will be given to it. After 7 days, it can be used to identify the malarial parasite. When the slide, by placing the spear, is rinsed with the methanol, after the few seconds or after the 5 minutes, we should place it in the Giemsa stock for 10 minutes, so that we can observe the malarial parasite when observed under the microscope. Next involves the spore staining. In spore staining, malachite green and safranin is used. Malachite green will give the green color, whereas the safranin give the pink color. Pink color is also known as the counter stain. It is the spore staining is used to stain the spores, whether these are the vegetative spores or endospores. Gram staining is used to differentiate gram positive and gram negative. Acid fast staining, it is used to differentiate acid fast and non acid fast. Giemsa staining, it is used to stain nucleus and cytoplasm, whereas the spore staining, it is used to stain the spores. The culture techniques. Culture media is used for the isolation and identification of pathogenic organisms. Liquid media and solid media are used for the cultural techniques. In the liquid media, liquid media is the media where in the liquid the test specimen or the microorganisms are placed. For this, nutrient broth, meat extract and peptone water for the preparation of sugar media and growth of bacteria are used. Nutrient broth is used for the growth of microorganisms. Meat extract and peptone is thoroughly used. In the solid media, it is used for the, we also known the EMB agar, McConkie agar, XLD agar, blood agar, chocolate agar, nutrient agar are the solid medias. Where the agar is used. Solid media is used to know the colonial appearance, hemolytical activity and pigment production. Biochemical reactions. Biochemical reactions are used to identify pathogenic organisms. These are as follows: sugar fermentation, production of indole, H2S production, methyl red reaction, Rogers reaction, oxidase test, catalase test, coagulase test, and urease test. Sugar fermentation. When there is the organism that involves in the sugar fermentation, then it shows the production of carbon dioxide and ethyl alcohol as shown in the first diagram. Whereas for the production of indole, if it is the indole positive, it shows the pink color precipitate, whereas the indole negative shows the yellow color precipitate as seen in the second diagram. Whereas for the H2S production, it shows the black precipitate formed is known as H2S positive, and for the H2S negative, no black precipitation is formed as in the third diagram. Whereas the methyl red and Rogers reaction, also known as the MRVP, MR positive and VP positive are in pink color, whereas the MR negative and VP negative are colorless or in the yellow color. In the oxidase test, after by the placing the bacterial culture in the oxidase test, we can observe the purple color if it is oxidase positive, no color if it is oxidase negative. For the catalase test, bubble formation involves the catalase positive and no bubble formation, it seems to be catalase negative. For the coagulase test, coagulation means clot formation is the clot coagulase positive and the no clot is the coagulase negative. For the urease test, purple color indicates the positive, whereas the yellow color or no color is indicates the negative. Serological tests. These are two types: direct serological test and indirect serological test. For direct serological test, we take the known antibody to detect the unknown antigen, whereas in the indirect serological test, we take the known antigen to detect the unknown antibody. For the direct serological test, it is used to identify of unknown organism, detection of microbial antigens by using specific known antibodies, serogrouping and serotyping of isolated organisms. Whereas for the indirect serological test, detection of specific and non-specific antibodies like the IgM and IgG by using the antigens or organisms. Next is the immunoserological test. These are heme agglutination, ELISA, latex agglutination, complement fixation, and immunofluorescent. Heme agglutination, as we all know, for the blood test we use the heme agglutination process. Now for the heme agglutination, when we use the antigen against the antibody present in our blood, then it shows the agglutination known as the heme agglutination. As seen in the picture, first one is no reaction, second one is heme agglutination. Next, for the ELISA, these are of three types: direct, indirect and sandwich. For the direct, antigen is placed to the primary antibody conjugate is used, then to this enzyme and substrate applied, whereas in the indirect, antigen and primary antibody to the Secondary antibody conjugated with the enzyme. Next, the substrate is added for the sandwich. For the antibody, antigen is placed onto the antigen antibody, and the enzyme containing antibody and the substrate is placed. Latex agglutination. For the latex agglutination, as like the heme agglutination, here the latex is used for the clot formation. Next, the complement fixation. In complement fixation, as seen in the diagram, antigen and complement are added in the serum with the antibody against antigen. Then the complement will fix. When we add the sheep RBC antibody to the sheep RBC, no hemolysis will occur. Then it indicates positive, whereas the hemolysis will occur, then it indicates negative. Immunofluorescence. We take the cell on that antigen is placed to that antibody is added, then fluorospore is added, then it is known as the direct, whereas for the indirect on the cell, antigen and primary antibody to that secondary antibody and the fluorospores are added. These are known as the immunoserological tests. Molecular biological techniques. Genetic probes, polymerase chain reaction, and plasmid profile analysis are used for the molecular biological techniques. For the genetic probes, DNA or RNA probes are used. Detection of a segment of DNA sequence in unknown organism using a labeled probe is known as the genetic probe. Means by using a labeled probe we can identify the unknown DNA sequence of an organism, whereas the polymerase chain reaction is the amplification of a short sequence of target DNA or RNA. After that, it is detected by a labeled probe. Next, for the plasmid profile analysis from the bacteria, we should isolate the plasmid and can be determined the size and number compared with the standard strains by axial electrophoresis. This is the genome inhibition, disk containing antimicrobial compound, microbes in agar medium. Minimum inhibitory concentration in this, as we follow the serial dilution on this antibiotic of known antibiotic concentration are placed in the test tubes within the patient's serum, or the antibodies present in the patient infection of the test specimen are placed in the concentration level as in the serial dilution. Then after incubation we can observe the minimum inhibitory concentration, means the concentration of antibiotic where it involves in the inhibition of organism growth. As we see in the picture, at 1.0 and 0.5 there is a microbial growth, but at 4.0 and 2.0 there will be no microbial growth, means it is the minimum inhibitory concentration of organisms. Whereas in the E test, on the agar surface inoculated with the bacteria or the test specimen organisms, E tests are placed, E strips are placed, and then it involves in the incubation. On the surrounding where it shows the inhibition, then it is measured on the E strip named as 1, 2, 3 on it, and then can be measured. Thank you for spending your valuable time on video. Thank you.